Here we have to pay attention to T cells potentially targeting each other and them, eradicating themselves as well as going after normal circulating T cells. And both of these can have serious consequences. And so, therefore, designing these therapies should be, a bit more careful. Now, we have developed several approaches that are targeting pan-T cell markers, CD5 and CD7, and both of them are quite effective in the clinical application. And we can generate these T cells that are resistant to fratricide. They don't really target each other. They don't get activated by each other. And the mechanisms for that are different. Actually, CD5 T cells, they're automatically resistant to fratricide. They don't need to do anything because the CAR is removing the antigen all the time, whereas the CD7, we have to either engineer them to become fratricide resistant to remove the target antigen, or we can expand them in the presence of pharmacologic inhibitors that block fratricidal CAR signaling, allowing us to expand and generate these T cells at clinical scale. And both of these CAR T cell products produce pretty good activity in patients with T cell malignancies, with T cell lymphoma and T cell ALL. And it's important to note that they're not just eliminating the tumor, but they also preserve normal T cell population and avoid T cell aplasia, which is very, very important in this application. So the safety so far has been acceptable, and we are looking forward to continuing these clinical trials and treating more patients to learn more.